It anyway. So advancing substrate-independent minds, that's what this talk is about, or what this workshop is about, really. So I'm going to go through a few basic things first. I'm just going to try to explain what that actually means. So what are we talking about here? And then why would you do it? And also, how would you do it? And of course, we can't get all that far with this sort of a very big topic in a small workshop or a small conference. But we can start working on some initial practical things, like what can we do now, or what can we understand about the fundamental problem. I can go right on to the next slide, in fact, if you can get them. Yeah. So, of course, I'm going to try to be within 10 minutes as well, so it has to be pretty quick. What we're all going to talk about here is really about how you take functions of the mind and put them in something else. Before we even start talking about that, you need to make a few assumptions. And one of those assumptions is that everything that is computable by a Turing machine. Okay, that's the simple way of stating the Church-Turing thesis. And okay, that's pretty universally accepted. There's also a strong consensus that what we have in our minds, what we are using to produce the functions of mind, what we're using to produce all the emergent things like our sensations, our feelings, our thoughts, that this is in fact emergent from biophysical processes, which we can also call computable processes, and therefore the mind is computable. So we have complex machines up here, but they're just machines. Machines in a certain substrate. We can go right on. Yeah. Now, what does that mean? We're going to talk about functions of mind and how to get them into another substrate. You've heard about something like this at the summit. At the summit, they talked a lot about brain emulation. We had speakers like Terence Sinofsky and Demis Hassabis, who is going to be here tomorrow, by the way, come and tell us some things about how you might emulate functions of the mind, how you might do that, and what you need to research in order to get there. So what is the level that you might have to investigate to get up to that point where you can implement those functions. In the previous times, there's been the use of the term uploading. I just want to bring that up right now because a lot of in the community know this term and are very familiar with it. It's a bit vague. A lot of people don't know exactly what it means. Are we supposed to be uploading or downloading? And are we just trying to get something onto an FTP server? So is this just, are we trying to get what is really a functioning human with thoughts and put them into a box and then just store them somewhere, like something you put on a disk? Or what does it actually mean? So it's a bit of a strange term, and we'd prefer not to use it. In fact, that's why we came up with the term substrate-independent minds, because this captures the idea, it captures the objective. We want to take the functions of what's going on in here, and we want to be able to put them in any substrate that can also compute the same things. But we don't necessarily say what that is or how we do it. Whereas, if we go on to the next slide, we have this concept of whole brain emulation, which has been discussed a lot previously also, and in fact, which is a term that I came up with originally, where you think, okay, the way to do this, to get the functions of mind into something else, is to try to recreate all the biophysical processes that are going on in the brain and put them into another substrate where you simulate or emulate everything that those processes are doing. That is not a bad idea. That's what I would call the conservative approach. That's the approach where you say, we don't know exactly what we're trying to copy. Let's just try to copy it as faithfully as possible and make it work the same way that it does in your mind right now, or in your brain right now. And then the idea, of course, is we try to re-implement exactly what's going on so that we can predict from one time step to the next. What should those functions be doing? What is the state of the mind? And, okay. Yes, please. Okay, this is something I just want to bring up because it came up during the summit. Right, so I believe, in fact, that it was Demis who pointed this out first, that we could look at the three different levels of organization according to Marr's method of describing them in terms of the computational, the algorithmic, and the implementational, where we'd say the computational is what is it that we're trying to do? What is the goal of these things? What are they trying to compute? What are you trying to end up with as a result? In our case, then, the functions of the mind. So everything that we experience or everything that's going on in our minds would be that goal. Or in any minds like that, let's say an incredibly intelligent artificial general intelligence or something like that.
Thank you very much.
Then the algorithmic. How do you do it? So what is your method of implementation? And one example of that is that you would do whole brain emulation. You want to recreate all of those functions as faithfully as possible, looking at the biology as the example. That would be one algorithm for that. Another algorithm would be to be more to use some kind of semantic process, more artificial intelligence, let's say. The implementational is then what do you actually implement it in? So the substrate, all these three levels. And during the summit, I think that Demis pointed out that yes, we have some ideas on the computational level, we have some ideas on the implementational level, and the algorithmic was kind of a problem. I think that ASIM of these levels, because we're saying we have a goal. The goal is that we want to implement functions like that. We want to be able to run that somehow. We want to be able to copy that or make a similar type of emulation. We don't say how, because we're not immediately jumping onto a technological bandwagon. We're being technology agnostic to begin with. But we also look at ways to implement. So what are the practical ways we can move forward with this? And let's go on. Okay, so now, how do you do that? Well, we think that it requires a lot of interaction between many different disciplines. This is something that you can't do if you're just a neuroscientist or if you're just a physicist or if you're, a chemist or whatever. So a lot of different people, sorry? No, okay. Yes. Anyway, so you need a lot of different disciplines, and this means that we need a way to get those all together. And usually what happens is when a lot of different disciplines need to work together is you start a new field of study. So this is how fields like neuroscience even emerged. I mean, they came out of different disciplines that already existed, and they sort of crystallized as a new field. So what I think is you need at least someone who's thinking about what are actually the fundamental components of this science. What are the basics that we need to understand? Where are we trying to head? What are the objectives? If you have an objective, then you have problems. And then you look for solutions for those problems. There has to be a structured approach. That means you need to build up a literature, and it means that you need to create a network of people. And for that, you need some sort of organization, or several organizations, in fact. And one of these, which is the face, I hope, for a lot of other organizations that will then link together, I hope, is Carbon Copies. So this is this group that we've started here, and you can see the t-shirts right over there, that is trying to do exactly that. And these events are part of it. So we've had the online events, and we've got this event. I'll try to go on. I'm going to have to speed up a little more, so obviously I'm taking too much time. Right, we'll just go straight through that because I already said it. Yep. Okay, a little bit about the mind or the brain. So why would you even want to do something like advancing substrate-independent minds? Why would you want to do brain emulation? One of the reasons is because computational limitations of the mind, for example. You might want to augment. You may want to have additional capabilities. We already use augmentations, of course, and that's also based on some of these limitations, like we forget everything. I forget everything I did yesterday. I don't remember anything. So I need tools to help me with that. I like to have long-term memory that is not finite and very slow at uptake of information. And I would like to have short-term memory, perhaps, that could have more items in it, so maybe then I could do more complex things. And my processing rate could be increased if I had a different substrate, for instance, etc. So you have all these different things right here that I'm mentioning on the slide, and more. I mean, there are other slides coming up, and I'm just going to mess it up a little bit by saying you could also want to be able to exist in a different place, for instance. Let's say you want to be in an environment that is not hospitable to our current biology, then you can come up with another form in which you could reinstantiate those functions of mind. So there are several good reasons to do this, and let's see if we can move through them quickly. I also believe that it is essential that we are able to augment and able to improve ourselves, because there is a competition that either we have with one another or that we have with machines that may at some point become very intelligent, very capable, and are able to take advantage of all these advances in materials and substrates and computational capabilities. So there's good reason to do this now and after the singularity, if we want to talk about that. And right, so now we go to the origin of it, which I'll also go through quickly. There is this connection with the neuroscientific interest in how to understand functions of the mind, and that's some of what you've seen during the summit when the neuroscientists were talking about it, which is we want to understand how the functions of the mind work. We want to understand more about what they now call the human connectome. There are some projects out there that are trying to investigate that. And yeah, so this ties directly into it. I mean, I'll show you a few examples in a moment, so we'll just go on. Here's the example, first example. Henry Markram's Blue Brain Project. Of course it was mentioned during the subject. It's one of the primary examples of new kind of neuroscience, neuroinformatics, where you really try to take the science to a computational point. It's like taking biology and taking genetics into bioinformatics. Here you want to take it into neuroinformatics. You want to understand more about how you can really work with the details of the functions that are there, predict things, make models, try out things, make experiments. And, well, Henry Markram's Blue Brain Project tries to do that by simulating a cortical column and running it on a computer. It's very similar to the goal of brain emulation. Really the only difference is this is a construct because this is created statistically from a whole number of rat samples and experiments done with those, statistics gathered about connectivity, etc. So you can say something about the general function, but it is not the implementation of a specific column. It's different than emulation in that sense. And I'll just kind of skip through this, but yes, Eugene Itzkevich did another wonderful interest model as well, where he kind of concentrated on size, as in how many things could he simulate very quickly, and then see if there was something that looked like the type of propagation of activity that you'd see in the brain. Also an example from neuroinformatics. Okay, there's a connection with artificial general intelligence as well. In artificial general intelligence, one of the reasons why you want to look at the brain as an example is because there we have a system that you can compare with, you can validate towards, and therefore you can ground your assumptions about how to achieve things such as the storage of new episodic memory, let's just say as an example. So in the human hippocampus, for example, we take on new memory, the hippocampus carries out certain tasks to do that, and then eventually those memories transition into the cortex at large. And there are some good examples there of how you could achieve this. And AI or AGI can learn from those examples. And in many different areas of the brain, you have the same thing, where you could learn from it and you can instantiate in a certain way that currently there doesn't seem to be any agreement in AGI, which, by the way, here seems to be AGII. Yeah, next phase, I guess. And we can go on. By the way, if anyone wants to throw out a question right away or something, I don't mind at all. That's fine. Now there's a connection also. So we've gone from neuroscience to AGI, and now we're going to the medical terrain, really, like how do you help people with problems. And in this case, it's Ted Berger with neuroprosthetics. I take him right away as an example because his is an example of a cognitive prosthetic, which to me is a whole different thing than, say, cochlear implants, because you're really trying to go right into the brain and see how do you do something in there that requires both input from the brain and has to give output to the brain within. This is an example of an attempt to look at part of a region in the hippocampus as a black box, understand what the inputs are, understand what the outputs are supposed to be, and then create the correct transformation. And he says that it works fairly well right now in experiments with rats, and he wants to move into primates to do the same sort of thing. This, of course, is a good way to fix parts of the brain that are very generic, and where you can just take out the broken part and put in a black box that does the same task. It is not a good way, for example, to fix memories that you may have lost, because memories are stored through the synaptic patterns in your brain. And you can't simply put in a generic part that knows nothing about what those patterns used to be and expect those memories to suddenly come back. So this is a very different thing than trying to fix, I don't know, someone who's had damage to the neocortex. Well, we can move on to that next one. Okay, so we need to look at not just why would you do it, but also how would you do it. That's really largely a topic for tomorrow, and I mean maybe a bit today, but it's a big topic because no one has really gone back and said, okay, we want to do uploading or brain emulation, and we want to lay down the fundamentals and explain what it is we need for this objective. How's that supposed to be done? So we really need to go back a step and define how would you even describe the kinds of functions you're looking for? How would you describe the state of a brain? How would you describe the transition from one state to another, or the changes in the brain when you're learning in a way where you want to take what is going on in a specific individual brain and put it somewhere else? So, for example, what is the resolution you need to look at? And that, again, was discussed to some degree in the summit. Do you need to look at spiking activity of individual neurons? This seems to be something that many neuroscientists feel comfortable assuming is the level to look at when we want to describe the state of the mind or a transition from one state to the next, because the neuron sees its environment in terms of, okay, spikes are coming in, my action potential raises, and eventually I spike. It's you can still debate it, but that seems to be the consensus. But then when you look at the change of synapses, for example, if you want to change these transformations in the brain, you might have to dig deeper. You need to look at the molecular level to understand exactly what's going on there. Is the strength of the synapse changing? How is it changing depending on the arrival of certain transmitters? And exactly how do you implement that if you want to do the same thing? Perhaps. I'm not assuming which level it is, but we do need to look at that problem. That's one of them anyway. Yeah. And the other big deal, of course, is that in the brain we always have this sort of mix of function and structure, just like we don't really easily explain what is the program and what is the hardware. It's not as easy to look at as perhaps in a CPU architecture. Both things seem to be essential, so the function of the components that are there, as well as the way that they're connected, the structure. And yeah, we need to understand more about that. Then there's the question, if you would like to do something like this, what is the kind of substrate you could put that into? What is the computational hardware that's required? What sort of computational capabilities do you need to have, in fact? And is it necessary to go further than the brain? A lot of what we do has to do with how we interact with our bodies, how we interact with the environment around us. So where do you stop exactly? What does it mean to capture who you are and put that into some other substrate? So that's the embodiment question. We can go on again. Right, I'm probably just going to skip this one. Yep. Yeah, I'm just going to quickly mention this because as you look at different possible solutions, how can you implement it, different technologies, you can go all the way from this whole brain emulation that you can just capture all of what you do on video, audio, write things down, get some idea of who you are, and then that perhaps could be a representation of who you are that would outlast you in some way. You would somehow become immortal in that sense. This is often called the Bainbridge-Rothblatt model, just because that's something that Martine Rothblatt is really pushing with the TerraSem Foundation. But it's not necessarily a bad idea. If we just think basics, okay, fundamentals here, you can, of course, always look at a set of data, and you can look at what is the average of that set of data. Okay, and if you take that average, you could abstract out something that, let's say, we could call the me program of a person. This is, what we all sort of have as the underlying structure of how do minds work? How does a human mind work? That's the me program. And then if you want specific me program, a particular individual, you would look for the difference between that average and who you are. Now that doesn't mean it's easily doable. It might be harder than doing whole brain emulation because perhaps the different me program or your differences are quite the same, it may not be so easy to do, but it's worth looking into if we go a little deeper perhaps than what the Bainbridge-Rothblatt method is doing. That's just my take on it, but I think it's worth going back and considering that as well. We don't simply dismiss things out of hand or jump on one technology. Just a very quick run over this one. This is a very typical example of an implementation idea for how you could do whole brain emulation as an instantiation of substrate-independent minds. It's the idea that we can look at structure and we can abstract from the structure what the function is. So we have both the Denk labs, the Hayworth-Lichtman lab, or actually Lichtman lab, but Hayworth is working there on an automated tape collecting lathe ultramicrotome, which is a nice long word for something that slices brains really thinly, puts them on a tape, and then perhaps even on silicon wafers so that you can start using the typical processes used in the chip industry, and then look at them under a microscope, which is something that some of us here are familiar with as a procedure for trying to solve biological problems, and try to read off what are these structures you see, reconstruct them in a computer, and then make a correlation between these structures that you've regenerated in 3D, their morphology, and the functions that you know from some library of all the components and how they would work. So in that case, you rely entirely on the structure to tell you about the function. And we can talk about why that might be good or bad later anyway. I think I have a slide about that in just a second. And in fact, not yet, but okay. The other way is look at just a function. Peter Passaro is Peter Passaro, who will be speaking here by the way. Oh yes, he's here. Okay, great. Hi, Peter. Will be talking to us about another approach, which is to look at in vivo data, to record from living brains or living pieces of brains, and in this case of Limnia, which is a simple organism, an invertebrate, but has, it has this wonderful feature that you can dissect it and it stays alive, and you can put it down on a microarray and you can record from it, and you can get a good idea of what's going on there, and then derive from all this activity not just what the activities are and how those particular components carry out their functions, but even what the structure or the functional structure between them is, because you can derive something about their relationships from the activity. So that's a completely different approach. Yes. Next slide, please. Okay, so besides talking about those technologies, you have to consider what these are missing. A lot of what brings about change that we can really work with, the change that is dramatic, revolutionizes science, is when we develop good tools. Tools are among the best things for scientists because they help us look at the picture in a whole new way. We would love to look at this particular problem by being able to look at large scale in the brain and high resolution, for example, but we don't really have the tools for that. And if we look a little bit beyond what's available right now, so beyond microelectrodes, beyond slicing things, we can think, for example, about nanotechnology or synthetic biology as ways of helping us develop tools that could look better at everything from the body to the brain and build machines, basically, that can help us out. One example is optogenetics, which is really the start of doing something interesting in that domain. In the case of optogenetics, it helps us stimulate inside the brain by delivering channels that will allow you to turn on and off particular neural cells based on a certain light frequency. And the reverse is kind of possible as well by making cells bioluminescent so that you can read out activity as they are activating from their luminescence. And yes, let's just go on now because I think I'm really out of time and way over time, in fact. Okay, so I'm not going to talk about that right now. We'll get to that when we talk about the... yes, that would work. Hey, look at that. That's much better. See, we learn as we go. I'll skip that one. Is this forward or backwards? Oh, that's right. Yes, of course. Yeah. No, it's because I'm right-handed, and I somehow assumed that the right button was, doesn't make any sense, does it? What's happening here? My mouse cursor is going all the way off. Oh, I have to put my hand under it. There. Okay. On. Yeah. I just quickly wanted to mention this, just because I was at a high-throughput microscopy meeting recently, and we kind of discussed a lot about the structural approach, because the structural approach is currently, in terms of technology, the most advanced one when you want to work in this area, with this problem, because we really can scan slices of brain at various resolutions that will help us see the cells, we'll see the axons, dendrites, and hopefully soon synapses if the atlum gets to a point where we can get large volumes out of it, because the atlum right now just works with small volumes. But there may be a problem there, because as I said, you rely on an inference between structure and function. And there are many reasons why you may not be able to simply rely on that. There may not be a one-to-one map between this structure and that function, or you may have this problem, the problem of open loop versus closed loop approaches. In an open loop like this, what you do is you try to create an entire implementation of the brain by mapping everything from the structure to the function using a library. And you hope that you didn't make any mistakes in your scan. You hope that you didn't misinterpret the thickness of the axon. You hope that you didn't misinterpret the function that you're trying to implement in any of the parameters. And when it's all there, you press go, and you hope that this thing works. That's not really the approach that you generally use in engineering. When you do something in engineering, you try to use a closed loop approach, where you get feedback all the time that tells you what you're doing and whether you're doing something wrong. So you're trying to validate as you go along. And yes, this was then discussed during that meeting, and indeed, there was suddenly an awareness that this could be a problem. Maybe we can't simply build the whole thing, press go. You need to be able to check what you're doing at every stage and see if it's still performing the same function, and then carry on and do the next bit. That doesn't necessarily mean that we throw away the structural approach. It just means we need to consider how to go about it. And what I'm showing there is simply the idea of the attractor network, because we think this is an attractor network, therefore you can do a lot of things to it and it will still give you the same result. It will still be able to recall the memories that you had, even if you shoot a bullet through it. But okay, sometimes this happens. Sometimes you can shoot a bullet through a head and the person recovers and they can still remember everything, because it is a distributed network. But it's not only a distributed network. It's a distributed network where different pieces depend on one another in a modular fashion. And besides that, if you do the scanning approach, for example, the structural one, you're not shooting a bullet through the brain. You're not just giving it some random destruction somewhere. It's not a random set of errors. Necessarily, it could be. But maybe you're making a systematic error, so that you're shifting everything right out of one of these minimum energy points in that landscape of attractors and into a different one. So it might not be the same thing as simply dealing with a distributed network with some random noise in it. Cumulative errors are important to think about. Yes? Well, one example I would think of, this is really Peter Passaro's approach, which is where you can watch what's going on. And if you have this snail, Limnea, on a multi-electrode array, and you can look at what those functions are that are being carried out and what the activity is in there, and then you do some of them in a computer so that you can replace this neuron and then that neuron. It's just one approach. I'm not saying that's the way to go, but that would be one way to test what you're doing while you're doing it. Okay. Yeah, okay, we just kind of discussed this already, and I really want to jump over it, so we'll go on. Yep. And that as well. We'll talk about some more of that later. I kind of mentioned a few of the next generation things. And so in summary, this is my summary slide. We're at the end. This is kind of the whole area we're going to talk about now. And I just want to emphasize again that we want to know what the objective is we're looking for. Advancing substrate-independent minds. I'm not immediately saying that we have to build minds the way they are now. I'm not saying that we need to stick to the rules of what is a human mind. We may look more at AGI, for example, as something to follow if it turns out that that's a good idea. But the basic idea is we can learn from biology, and from that we can build something that we can test, that we can validate, and that works. And we want to be able to re-implement the functions of the mind. So how do you do that? You need to understand something about the fundamental problems, and then you need to look at what are the possible solutions so that you can build a roadmap of things to do now, actionables. A roadmap can never be something complete from A to Z because we just don't know enough. We don't understand where we are, and by the time we get somewhere along this roadmap, it'll be different. So we need something that's dynamic. That means we need an organization that will look at all these possibilities, see what kind of results are coming up, keep looking at the big picture, understand if this is getting us to the requirements that we need, and keep on adjusting. So that's just basically what any field science does when it's properly organized field of science, which has journals, which has conferences, and a lot of people organized in it who know what they're after. And that's pretty much all I wanted to say to introduce this. And you had a point. Yes, why not? Yeah. It would be a substrate-independent mind. An AGI like that would be a substrate-independent mind, yes. But I'm not saying that it couldn't be a specific human mind. There's no, those could both be substrate-independent if you learn from the human brain how to make those functions work and implement that. So, yeah. Yes. Oh, I'm sorry. Yes, I'll do that. Okay. The question that was posed by Raman Sam, if I'm pronouncing that correctly, close enough, is the goal? Does the goal include emulating specific individual people's mind? So, is it the uploading meme, basically? And my answer was, it doesn't have to be, because you can also make substrate-independent minds that are really different minds, like say some generic instantiation of something that's based on human brain function. But yes, that is also part of the goal. I mean, the idea came originally from whole brain emulation, which was about how do you as an actual implementation. So my answer would be, generally, yes, I would seek to do this for individual human minds, but it doesn't have to be, because advancing substrate-independent minds has more connections. It has a bigger, let's say, scientific playing field. Okay. So, you mean like a hierarchy between the general human mind and an individual human mind? Yes. Oh, okay. To substitute these functions to make sure that are properly. Okay, I'm going to try to relay this, what we're talking about. And Leslie Seymour, right? Okay, your question was, there's a whole range between having a generic implementation of a human mind and having an individual person's human mind. Could you take functions from the generic version, for example, to fix something in an implementation of an individual mind if there was something wrong with it, or you didn't know if it was correctly copied or something like that? And I mean, this is, of course, exactly the same as doing what Ted Berger is trying to do by taking a generic implementation of a piece of the hippocampus to fix the hippocampus in people who've had a seizure and a part of their hippocampus doesn't work anymore. So yes, you could do that as a medical procedure the same way you could in a biological person. Yeah, that's my answer, really. Oh. So Suzanne has left us temporarily. Yeah. Now, the parts of the roadmap that exist currently consist of the structural approach, which was largely laid out in the whole brain emulation roadmap that was written up in 2007 at a meeting in Oxford. There's also some work in there that discussed the functional approach, but it was sort of biased towards the structural method. And in fact, left out a lot of other possibilities at the time. So it was really whole brain emulation specific, not substrate independent, mind specific in that sense. What we do have in terms of a roadmap for the general case is some idea of how you might represent the basic functions of the mind. I mean, what kind of representation could you use to talk about what sort of functions, parameters do you need to extract, what sort of data do you need to extract, how would you even model that? And we have some notion of the different areas in which you can look for solutions. So a functional approach based on multi-electrodes, on neural implants, on more synthetic biology approaches, nanotechnology approaches. So we have an idea of where to look. And so what we really have is the objective, a few possible routes that are kind of mapped out, and this notion that we need to organize it better and understand more about the fundamentals to move forward. So that's where we are. Yeah. Okay. Yeah, that's a really good question. In fact, I think that's a perfect spot to start the workshop in terms of people discussing this. And so I wish I had a whiteboard right now because I would like to put that up there. How do you measure progress along a roadmap or along understanding how to do substrate-independent minds? Anybody got any ideas about that besides myself? Sorry? Mm-hmm. Yeah. What do you mean by numbers? I think you do, actually. I mean, I've looked into it quite a bit, so I know that, yes, indeed, if you look at what Henry Markham's done or Eugene Skivich has done or Dharmendra Modra has done, that all these people have made large simulations of brain areas or the entire brain, trying to replicate microcircuits to a certain degree. But always you see that they are getting results that are usable in terms of testing them against evidence from the human brain, such as they see oscillations that have the right frequencies, or they see patterns of activity that are propagating the way you would see it in an fMRI, for example. So there is result in that sense, that the neuroinformatics is getting better. There's also result in terms of the ATLAM, for example, or the knife-edge scanning microscope as examples of tools that can help you collect data that is both large scale, as in we try to do this for entire brains, and at high resolution. So there's a way to do that, at least for the structure parts. And now there are also some tools on how you might be able to detect chemical content better and things like that. So their tool progress as well. Yeah. Yes. Mm-hmm. Mm-hmm. Yeah. I would say that the level of understanding is certainly not complete, but it's This is one of the things that you can approach by, for example, the type of studies that are being done in vivo as well as in culture, where you can look at the system and you can tweak it and test it in various ways. So this is really a case of trying to collect a large number of data points for all these different kinds of parameters that are available. It's the kind of stuff that, in fact, they do at the Blue Brain Project, for example. They do a lot of work there trying to collect that sort of data. It's the sort of thing you would do if you're trying to reverse engineer any system. Because, I mean, you could change the temperature, for example, in the brain. That will change a lot, just like changing the dopamine level would. And you can do the same thing in this computer. If I change the temperature a lot, then that's also going to do a lot. So clearly this is the sort of thing where you need to map out what that does. And, yeah, that's not complete at all. Yeah. Yeah. Mm. An emergent phenomenon. Oh, sorry. Yeah. Yeah, that's difficult to grasp, really. Yeah, I think that I mean, right now there's so many problems on the practical level just to understand how this machinery works that mostly I focus on that. I mean, when I think about what are the problems that we need to solve. Yeah, okay. So Peter Bossaro, for example, is trying to tackle exactly this problem. That's good to know. Yeah. So yes, I'd like to know what you have to say, but I just want to bring up once again that I'm really interested in answering the question that Mark posed, which is how do you measure progress in this? So I'd really love it if people come up with some ideas. There, you have an idea, and is yours also an idea for that? Okay. So, well, okay, great, lots of ideas. Is anybody taking notes on that? Yeah, you're kind of relaying what we're saying here, aren't you? Yeah, so then by doing that, you're actually noting everything that they're saying. That's good. Yeah. Oh, yeah, just give them the general gist of it. Yeah. Okay, you first, David. Hmm. Hmm. So basically, if you get the behavior right, and then you check what is in this organism, what is it that you have set up in your emulation, then you can get some kind of a metric on that. Okay, that's. Hmm. Okay, and you had an idea too, Max. Right. Right. Yeah. How do you measure how much it is an engineering problem versus a scientific problem? Yes, it definitely has an end deliverable, yeah. Hmm. Yeah. Right. Yeah. 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 Kind of related to that, I just want to mention, if we're trying to measure how far along are we to having a substrate-independent mind, I might say something like, okay, we're all the way there if we basically have what Ken Hayworth and Peter Passauer are looking for, which is a way to transfer an entire biological brain into another substrate where it functions. But if let's say I'm completely dependent on a number of augmentations, and some of them are, say, neural interfaces that I've got, and they offload some of the images I see every day, something like that. They help me with my memory. They give me access to Google and Wikipedia right away and stuff like that. So it becomes a part of me, and I'm sort of offloading part of who I am onto that. Am I 50% of the way there, or 25% of the way, or something? This is sort of also a measure of how far you are towards being some other kind of substrate. Not quite sure how you would measure how far you are, but it certainly does seem to indicate a type of progress that is, I think, a likely sort of progress to see, just because even when we're discussing here as an interdisciplinary problem, we're still, which is that there's a whole other set of fields out there like augmentation, VR, etc., that are going to make progress as well while this is going on. So clearly you're going to see a lot of interaction between these different fields and different interests, and you'll see progress that sort of walks along in a kind of Brownian motion with all these different things going on at the same time. Any more ideas there? Yes. Yeah. So what is farm? Yes. Well, yes, I'm going to comment on that as well, because David said there's been a lot of progress in this because the general state in the brain based on things like neurotransmitter levels is something that pharma is really interested in. And the truth is, of course, that yes, pharma has these drugs work for some people, don't work for other people, may not work that well, have lots of side effects and things like that. So even though it's interesting to pharma, it's definitely far from solved problem that they are interested in that. And you had, yes. Mm-hmm. Right. Okay. Yeah. Mm-hmm. Yeah. Okay. Yeah. Okay. Those are very relevant, very fundamental questions. But of course, if we have measures for them as well, that would be really good. If we know how far along we are to answering those questions, would be really great. Yeah. 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 Okay. I need to do a moderator thing here, and I have to say that because of the time and because we have some other topics coming up, I'm going to ask for one or question, and then we should really move on to the next bit or session here. Yes. Yeah. Yes, it's going. Yeah, yeah, that's right. Right. Mm-hmm.
So how do you communicate it to computer scientists?
Mm-hmm.
How do you communicate it to lay people? And then this is a medium for presenting.
Mm-hmm.
So
Mm-hmm.
that's a bit in preparation for a workshop like this. People walk in with the knowledge that they need to see the totality.
Yeah. Yeah, I think that's very important to at least have, say, for example, a literature related with that or media that of some kind. But not just that. I even think that projects that spawn off of the research that's been done, say something that is immediately implementable, maybe even commercial or anything like that, is also a way to bring that forward, to present what you've achieved. So I think, yeah, that's a really good point.
Then extension of that is